We will talk about the Human Genome Project. Human genome is consisting of 22 autosomes, X chromosome plus Y chromosome plus M chromosome, consisting of 25 DNA molecules. Any genome is equal to total DNA sequence of the particular organism. In this human genome, it is sequenced totally, consisting of 3 into 10 to the power of 9 million base pairs. And after the sequencing, it is necessity to construct a maps. Generally, the maps, which is consisting of two types, genetic maps and the physical maps. In case of the genetic maps, they are the rough maps. We can use the two alternative forms of the any gene. For example, in case of the Drosophila melanogaster, we have the two genes on the second chromosome consisting of normal wings and the vestigial wings on one locus in the form of the two alternative forms of the phenotypes. In the other locus of the same chromosome consisting of body color black and the gray color, and we can construct a genetic map by taking the crossovers percentage by the total progeny. We will get the crossovers ratio and multiplied with the 100, it gives the map distance in between the two genes. So genetic map is units in the form of the centimorgans, consisting of 1.2 million base pairs or 12 lakh base pairs. So anyhow, genetic maps are very rough maps. It is possible to construct genetic maps up to the number of chromosomes, maybe 23 genetic maps are possible in case of the humans. But we need fine maps, that is in the physical maps. In addition to that, we have the restriction maps by using the restriction enzymes. Restriction enzymes also not solve the problem, and further it is replaced with the RFLPs, restriction fragment length polymorphism. It is earlier used for the disease diagnosis, later it is used for the construction of the maps. Further, it is replaced with the physical maps. In case of the physical map, we can take a distance of 1 lakh nucleotides, and we can take the left and right margins of the each 1 lakh nucleotides, and we can construct the physical maps in the form of the certain sequence length of the DNA. And we have the 30,000 markers in the human genome into 1 lakh. It gives rise to 3 into 10 to the power of 9 base pairs or the 3 billion letters. With the physical maps, we can identify the any region of the chromosome with no time, and we can sequence very easily. And also the human genome sequenced very quickly done, because the 4-lane method, it is replaced with the single lane method by using the capillary electrophoresis, and also replacing the radio labels with the fluorescent dyes. Because of that, the human genome sequencing is carried in a very quick fashion. In addition to that, the earlier human genome is sequenced by the hierarchical mapping by knowing 1 lakh fragment in the left and right margin, and they are broken into 500 base pairs, and it will be sequenced and it will be aligned. It will a time taking process. It is replaced with the shotgun method of the DNA sequencing by breaking 1 lakh fragments, not knowing any left and right margin. For example, in case of the Haemophilus influenza, it consisting of 1.8 million base pairs, and it is broken into small fragments, and it is sequenced 4x times and aligned and sequenced in a shotgun method, very quick fashion. This is implemented in case of the other organisms like yeast, C. elegans, and Drosophila melanogaster, and later on it is implemented in the human genome also. Like this, the human genome is sequenced very quick fashion, and it is useful in the public domain. Anybody can access the human genome. So altogether, the human genome consisting of, earlier it thought that 1 lakh genes. Later it is condensed into 50,000, after that 35,000 genes. Now it is considered only 25,000 genes. But number of genes is 25,000 genes, but gene products may be equal to an average of 4 gene products for a single gene, and we can get the 1 lakh peptides. With this human genome, which is given a thought provoking, and by sequencing other organisms simultaneously, the human genome which is sequenced thoroughly, it may be useful further in the analysis of the different kinds of the diseases and other things. Thank you. Human genome further analyzed consisting of only 1.5% of the genome is expressing. The rest of the genome is silencing. And we can see the 75% of the genome in intergenic region which is not expressing, and the remaining genes are occupied in the 25% of the genome. Only the genes are expressing in the form of the exons in the 1.5%. Within the genes, intragenic regions, we can see the in the form of the untranslated regions in the form of the introns. So 23.5% of the genome is not translating. Like this, we can see the only 1.5% of the genome which is transcripting and maybe translating. The rest of the genome is silent. And also the human genome consisting of satellite DNA in the form of the highly repetitive DNA in the form of the STRs, simple tandem repeats, in the form of the microsatellites consisting of 80 rich regions, 1 to 5 nucleotides. It may be forming in the human genome, while DNA duplication, the DNA polymerizing flips out from the template DNA. After that, again it joins with the template DNA. So in the next duplication, we can see the loop-like region which is duplicating. It is forming the simple tandem repeats. These simple tandem repeats are causing certain diseases and vibrations in the humans. In the shaking condition, we can see these simple tandem repeats, which are useful in case of the DNA fingerprinting analysis. In addition, we can also see the VNTRs, variable nucleotide tandem repeats. The length of VNTRs may be in the form of the 1 to 50 bases, 80 rich regions. These VNTRs might be forming by unequal crossovers. So VNTRs earlier used by the Lake Geoffrey for the DNA fingerprinting. Later it is replaced with the STRs for the constructing of the fingerprints. And also we can see the human genome, which is occupied by the 50% of the genome with the transposable elements in the form of the lines and the signs. Lines are the long interspersed elements, and signs are the small interspersed elements. So lines are forming with the retrotransposable elements, which are synthesized by the RNA polymerase II, and whatever the RNA is converted into DNA, and it will be incorporated in the human genome. And we can see the signs of the length of maybe 300 base pairs, whereas the lines may be 6 kb or 7 kb length. The signs which are synthesized by RNA polymerase III, and it is incorporated in the form of the simple and in the form of the signs. So like this, we can see the the transposable elements which are occupied in the human genome near about 50% of the genome. In addition to this, we can also see in the human genome in the form of the pseudogenes. The pseudogenes are nothing but whatever the RNA is transcripted, mRNA, which is again inserted into the human genome in the form of the pseudogenes. So like this, altogether the human genome which is analyzed, only 1.5% of the genome is expressing, and we can see the 75% of the gene region of the chromosomes not having any genes, and remaining 25% regions of the chromosome occupied with the genes. Within the genes, we can see the 23.5% of the genome is not translating. Like this, we can see the human genome, and it is occupied with the microsatellites and the mini satellites in the form of the STRs and VNTRs. In addition to that, we can see the lines and the signs in the form of the retrotransposable elements. And in addition to that, we can also see the 10% of the genome is occupied with the pseudo genes which are formed by the RNA conversion to DNA. Overall, the human genome, which is containing 22 autosomes plus X chromosome plus Y chromosome, a mitochondrial chromosome, that is the M chromosome, altogether it is consisting of 25 DNA molecules, and the length of the genome is 3 into 10 to the power of 9 bases, that is the 3 billion letters, and it consisting of 25,000 genes and the gene products in the form of the 1 lakh peptides. Like this, the human genome, which is analyzed and sequenced thoroughly, and it also useful to analyze the other genomes. Okay.